Good morning, everyone. Today, we're going to study the vestibular system. What we would like to accomplish first is to understand how the vestibule looks like and the relationship to its function. The goal is to learn how an animal can do this type of action, that is, to maintain balance.
Unbelievable.
To succeed in the above objective, we need to comprehend the mechanisms an animal uses to recognize its posture and any movement. Finally, to relate these subjects to your future activities as clinician, we will mention a common pathology associated with this system. Let's see how the vestibular system looks like. You remember the bony labyrinth? We introduced it in the last class in relation to audition. The vestibular system is divided into two functional components. The static labyrinth, which contains two structures, the utricle and the saccule, encapsulated in this area which is called the vestibule. The function of the static labyrinth is to determine the position of the head and to detect linear acceleration or stopping. In terms of head position, the static labyrinth recognizes when an animal is standing normally with the head up. It recognizes if the animal has the head tilted to a side or if it is laying down with the head flat in the ground. In summary, recognize the position of the head with respect to vertical and horizontal position. The other role of the static labyrinth is to recognize linear acceleration or deceleration. Recognizes when the animal moves in a linear manner. This could be walking forward or by being carried in a trailer. As it recognizes acceleration, it also recognizes when the movement is slowing down and eventually stopping. The other component of the vestibular system is the kinetic labyrinth, which consists of three semicircular canals. Each semicircular canal is positioned perpendicularly to each other. The role of the kinetic labyrinth is to keep track of the rotational movement of the head. All movements such as up and down, as if nodding, or sideways, as if shaking the head, or tilting the head to one side or the other. Looking in a bit more detail at the vestibule and the semicircular canals, and opening some virtual holes towards the interior, we find that in the vestibule, the utricle and the saccule contain certain similar structure but positioned in a different manner. And we also can identify in this image that each semicircular canal contains an enlargement called ampulla that also contains some special structures. If we further magnify the vestibule, we can now recognize that both the utricle and the saccule are located mainly within the membranous labyrinth, and each contains a small patch full of hair cells, which is called a macula. We also find that the utricle sits horizontal in the vestibule while the saccule is placed vertically. If we peek into the ampulla, we also can see that there are some hair cells, but in a different configuration. Before we move into the functioning of these structures, let's briefly review again the basic sensory cells of these organs, the hair cells. The hair cells in the vestibule system are a bit different than those in the cochlea, but they both operate under the same principle. In the cuticular plate of the cells, there are several rows of stereocilia of different height that finish in the tallest structure, which is called, in this case, the kinocilium. The kinocilium differentiates vestibular cells from those in the cochlea, but there is no different function identified for this taller structure. These finger-like structures are bound to a spring-like connection, as you remember, which connect the shorter stereocilia to a spring-gated potassium channel located in the taller stereocilia or in the kinocilium. In the body, the hair cells has voltage-gated calcium channels as well as potassium leak channels, and each cell is connected to a ganglion cell which goes to form a vestibular nerve in charge of carrying the information of each cell to the higher centers for integration. Going back to the utricle and the saccule, here we can see that each of the macula, which is a patch of hair cells of about 2 millimeters in humans, are placed perpendicular to each other. The macula of the utricle is horizontal and the macula of the saccule is placed vertically. In the next view, we can observe that each macula has a kidney-like shape. But the most interesting feature of this structure can be appreciated in this image. If we look with more attention, we can recognize that each hair cell is oriented in a slightly different angle. Not two cells are pointing in the same direction. As a reference, I suggest that you identify the fattest kinocilium of each cell, and you see that they are all point to a slightly different orientation. The idea is that all possible orientations are represented within each macula of both the utricle and the saccule, and this will contribute to the proper function of these structures. Furthermore, all hair cells of each macula are covered by a gelatinous mass-contained denser structure made of either protein or calcium carbonate called otholites or statoconia. Because of the higher density than the medium in which they are found, the gelatinous mass, the otholites respond to gravitational forces by moving the entire gelatinous mass which cover the hair cells and deflects the stereocilia and kinocilium in a given direction. This picture represents more realistically the structure of these maculas, whereby only the stereocilia is located within the membranous labyrinth, thus exposed to endolymph. The body of the hair cells is embedded or supported by a variety of supporting cells which are located within the area of the bony labyrinth, which is exposed to perilymph. Remember how these cells work, and this will help to clarify why this happened. The macula of the utricle and the saccule are covered by a patch of hair cells, each in a slightly different orientation. The tip of the hair cells are covered with and in contact with a gelatinous mass, which is embedded with dense structure made out of proteins and calcium carbonate, known as otholites or statoconia. The combination of gelatinous mass with heavy particles incorporated permit that this mass, following the law of inertia, tend to stay in place when the heads start to move and tend to keep traveling when the movement of the head ceases. As a consequence of this phenomenon, the stereocilia and kinocilium initially bend in the direction opposite to the initial movement, catches up and straightens during the duration of the movement, and when the movement stops, this must bend the stereocilia and kinocilium in the direction of the movement. Since all stereocilia and kinocilium in the macula bend in the same direction, but each cell is oriented in a different manner, then a given movement may result in the full depolarization of some cells, in partial depolarization in other cells, and in full hyperpolarization on others. The opposite occurs when the movement stops. Since the central nervous system recognizes the position of each cell, the central nervous system receives all the information of a given movement, and it can compute and interpret it as such. In this manner, the central nervous system can identify movement on any direction of the 360 possible degrees, as well as the speed of displacement. In the kinetic labyrinth, the sensory structure is located in the enlargement of the semicircular canal called the ampulla. The structure is called the crista ampullaris, and it is made of a series of hair cells aligned in the same plane as that of the semicircular canal. The stereocilia and kinocilium of those cells are within the membranous labyrinths. Therefore, they are bathed in endolymph. The body of the hair cells is located outside the membranous labyrinths in the volume filled with perilymph. The tip of those cells is covered by a gelatinous mass called cupula, which, at a difference from those in the sacral and utricle, do not have ocellates or statoconia. The cupula, however, attaches in at the tip of the membrane of the membranous labyrinths. This design creates a sort of blockage in the lumen of the membranous labyrinth component of the semicircular canal, which will permit the proper displacement of the cupula in order to carry out the operation of the crista ampullaris. We will review this shortly in a small video. Three semicircular canals are responsible for detecting the rotation of the head. They do this using a sensory organ called the crista ampullaris, which is located in an enlargement of the semicircular canals called the ampulla. The crista ampullaris also contains hair cells, but unlike the macula, the hair cells in the crista ampullaris are all positioned in the same direction, lined up with the plane of the semicircular canal, and their stereocilia are covered with a gelatinous mass called the cupula. The endolymph, which fills the membranous labyrinths of the semicircular canal, moves the cupula to bend the stereocilia in only one direction, which in turn depolarizes the membrane and creates more frequent action potentials. This is possible because as the head moves in one direction, let's use the horizontal semicircular canal, which detects sideways movement, the inertia of the endolymph tends to keep the fluid in place, thus pushing the cupula in the opposite direction to the movement of the head. Because the plane of the other two semicircular canals is perpendicular to the direction of the movement, the cupulas of those crista ampullaris are not affected by this type of movement. If the movement continues, then the endolymph reaches the same speed as the movement of the head, thus stopping the signaling. As soon as the movement of the head diminishes, the inertia of the fluid bends the hair in the direction of the initial head movement. This causes the hyperpolarization of the hair cells. If we look at the activity of the hair cells in the vestibular system or in the ampulla, based on the frequency of firing, we find that while the stereocilia is standing up, the cells fire at its resting frequency. That is, a frequency that will not trigger a depolarization. When the hair cells bend in the direction of the tallest stereocilia, and the tip link are able to open the potassium-gated ion channels in the tallest stereocilia, the increase of intracellular potassium makes the cell more positive. Therefore, it fires at a higher frequency, triggering a depolarization event. And when the movement catches up and the stereocilia straighten up, then the firing returns to the frequency with which the cell is in the resting mode. But when the movement stops, the cupula or the ocelot will bend the hair cells in the direction of the smaller stereocilia, triggering the low frequency of firing characteristic of hyperpolarization, which will last until the movement ceases and the hair cells return to its standing position, thus generating the resting frequency of firing. In terms of pathologies caused by deficiencies in the vestibular system, the most common problem is that of vestibular disease. In this short clip, you will see Marty having the head tilted towards the left, and when it walks, it tends to stumble towards that side.
Come here, Marty. Marty.
It doesn't have a very severe
Come here, girl.
case, but it
Marty.
impairs his quality of life to a great extent.
Look how much
Although
your head's
this
laying
animal
down.
still keeps a very good humor.
Come here,
and tries to
Marty.
play with a
Come
little
here, girl.
toy and
Come
stuff.
on.
There you go. The problem,
Back left leg.
which is more common in advanced age, is caused by many reasons, with infection in the inner ear is one of the most common. But the disease is usually diagnosed as idiopathic. What we see in an animal suffering from vestibular disease is, as you saw, the tilting of the head to one side and stumbling as if it is falling. It is believed that the reason for this is that because the balance system is not working, the animals appear to perceive that the horizon is inclined, and in an attempt to see it flat, the animal tilts his head. Because the animal perceives the horizon abnormally, the animals attempt to compensate for that deviation and incline itself to the opposite direction to the point that they lose balance and almost fall. And in many cases, they do fall. Always the inclination and the fall are toward the same side that the head is tilted. A most subtle observation is that the animal manifests nystagmus or jerking of the eyes. In the clinic, you will learn more about this abnormality as well as to potential treatment. Well, that's all what we have for today. I promise you a shorter session, and this is what this one is. To summarize what we have done is we went into the structure of the vestibule with all its components, followed by revisiting something that we have learned in the last session about how the hair cells function. Then we discuss in a little bit more detail how the macula of both the auricle and the saccule function, as well as those cells in the crista ampullaris in the cupula. And at the end, we view a little bit of a common pathology associated with deficiency in the vestibular system. I hope you enjoyed it and have a good day.